in our NGSeminar series. This week, Michał Krassowski is going to talk about how to up our data scientist game by using computational knowledge. I am pleased to announce that the next week we're going to continue on this road with a workshop by Edgar Garriga about creating a reproducible scientific workflows using Nextflow. So a couple of announcements. Here in NGSchool Society, we are working hard to bring you seminars and a virtual conference in the future. The schedule for webinars is available at our website atngschool.eu. For people watching on YouTube, I greatly encourage you to join our Discord server and participate in exchange of knowledge between computational biologists and trainees all over the world. And you can find all the links in the video description. Okay, so we have some slight technical issues. I think our speaker got disconnected. But this is for our announcement, and we're going to start in a moment. Oh, great. All right. Yes, I think we can. See you.
All right.
Okay. I think we can hear you too. So yeah. Okay. So we are battling some technical issues, but I would like to introduce our today's speaker, Michał Krassowski. Michał is a PhD or DPhil student at Oxford University, and his research focus is employing molecular data integration to discover the molecular basis of endometriosis and other chronic pelvic pain conditions. He holds a bachelor degree in bioinformatics and system biology from the University of Warsaw and a master degree in biomedical research from Imperial College London. He has a great deal of programming experience, which includes contributing to scientific software development initiatives such as Jupyter Lab. So, Michał, welcome. The floor is yours.
Hi, everyone. To introduce you to computational notebooks in the context of biomedical research today, and level off with explaining what I mean by computational notebooks. And basically, there are two terms, two constructs, which are sometimes called Jupyter. I will be mostly focusing on the Jupyter notebooks, which include text, code, and interactive visualizations or any other data output, which is also saved within the notebook, as in contrast to our Markdown or our notebooks, which also can display interactive visualizations. However, until exported to another because these slides will be uploaded to the Jupyter GitHub repository, which I think was already shared with you. Okay. An example notebook which explores the PTM modifications and gene expression regulation in. You can see that it includes both a lot of texts which explains what the research is about and code which text and produces the actual analysis of the more interesting feature. Heat map visualization, which if you is interactive, however, we may need to wait a little bit until it is fully loaded, which greatly improves the ability to identify specific values that were changed, adjust options, and explore the data in much greater detail than it would be possible with a static heat map, which was the standard for many, many years. So while this is nothing new, the ability to share such an interactive visualization with the world as an additional interactive publication possible static heat map, which was yes. So while can I just ask the team to share such an mute themselves because I'm hearing myself in a loop. Interact. Okay, great, thank you. The other example that I wanted to highlight is demonstrating how to that we can also visualize a higher number of dimensions, and it is in context of Zika virus analysis. So here the authors explored different PCA components and they were able to share a 3D PCA plot with us, which is also available online using Plotly. And finally, this paper, which explores evolutionary distances between sequences. Here I just wanted to highlight that various formulas can be embedded in Jupyter notebooks, as well as in other notebooks, which is a very useful thing when writing, presenting scientific results, which can be much easier explained together with this scientific formulas embedded rather than written in a plain text. So I would encourage you to check out the other examples in there. Now onto more details. So R Markdown or R Notebook, which are constructs from the editor, which is called RStudio, and you already have had a chance to work with that on the previous webinar, are almost identical in this context. And they are basically Markdown documents, so text documents with code embedded within. The outputs are not saved, but they can be saved when exported to a number of different formats. And these are strictly focused on R. However, other languages can be also used with R notebooks or R Markdown. For example, Python can be accessed or run via special package that you need to install. And while it is tightly tied to RStudio, it can be basically viewed in any Markdown viewer. And there are some other code editors which also support R Markdown. And it provides support for widgets, which I will talk about later. Jupyter Notebooks, on the other hand, are not text files, they are JSON files which need a dedicated viewer and are much more complex inside. They were designed to work with multiple different languages, which are technically different kernels providing support for specific languages, and for any given language there might be multiple kernels providing slightly different variation of support for this language. And the very name of the Jupyter project comes from Julia, Python, and R. And at the moment, there are over 140 different kernels registered on the Jupyter project website, and possibly even more in the wild. So there is a great variety of support for whichever language you would like to use. And Jupyter Notebooks also include multilingual cells, so you can mix and match different languages within the same notebook. The export options are also quite diverse, and those are supported in many more editors. However, there is, so I just highlight that there is Jupyter Notebook and Jupyter Lab. Today I will be showcasing some features of Jupyter Lab, which is a new interface for Jupyter Notebooks developed by the team with a large overlap of the team book originally. So as I mentioned, both are Markdown notebooks and Jupyter Notebooks support widgets, which will be explored later. For notebooks, many of which are commercial or developed by high-profile companies. For example, Netflix wanted to have notebooks which have even better support for mixing different languages, so they created Polynote, which was very well. There are many Jupyter-based solutions that you might have used, or JetBrains, Datalaw, Microsoft Azure Notebooks, which this one actually will be closing down soon because Microsoft is exploring yet another solution, and many more. The final one we have notebooks which actually predate the Jupyter project itself, so that they were the original inventors of the notebooks. Has become very popular over the recent years. And onto a short view of the features that you can access. As mentioned before, you can have equations, and those are embedded in Markov cells with, as you can see here, I would appreciate if someone could confirm that the window that I'm sharing has changed to the Jupyter Lab interface.
Yes, it has changed.
Okay, thank you, great. So you can see that with a notation of double $sign, we can create an equation which is centered, and with notation of a single one, we can just embed it with an okay. And then to end the work with mathematical symbols, or you can quickly type the name of the symbol you would like to use and press Tab. And because I am currently in a Python notebook, which you can see right there, I can use it as a variable. It will work equally well as if I just typed A. But because of the additional often match the current use in the literature, I'm able to represent my intent better with those symbols. Then further on, we can embed various plots. To make this example a little bit more interesting, we can add more elements and plot a histogram like that from Pandas series. I will just remove this semicolon for a moment so you can see that it is a nice trick to hide the representation of the plot object. So if you are getting any extra text, you can just add semicolon and then it should disappear. Then onto the widgets, and specifically to the widgets which are useful for biomedical research and research in biology in general. FASTA, which is a popular format among bioinformaticians, has a dedicated viewer. So here we have some assembled genomes of Zika, and this is just the file which is stored here, and I could open this in a simple editor, but it's rather suboptimal to explore and not very useful in this way. However, when using the built-in feature of FASTA viewer, we can interactively browse very large genomes together with the alignment if provided in such a form. Another useful widget would be to display three-dimensional data. So here, using ITK widgets, I loaded an image of a brain of someone who's very young, I guess. And you can explore it interactively and also publish for others to explore. Then you can have your own custom widgets, so you can create as different sets of controls to manipulate the display of, for example, your function, as demonstrated here. So we have an equation which multiplies our x by m and adds b. And if we change m, we expect that the slope will be changed. And if we change b, the position will adjust accordingly. And the ranges of the possible values are provided here as a parameters. So we cannot exceed -2 or 2 for m. And the third parameter is the increment, the step. So we are only moving by 0.5 in this case. So as with everything, the link to the IP widgets documentation will be available through the slides, from the slides which will be uploaded, and I encourage you to check that out because it provides very easy introduction to writing your custom interactive widgets. Let's go back to slides for now. So in addition to widgets, some languages have additional support for interactive use of various features, which includes integration with your operating system. So for example, we can execute commands in Bash. Because I'm currently using Linux, it's very easy for me to, for example, check what is my current location. I create a test file, which is named test.txt, and its content is just test. And the only thing needed here is to add the exclamation mark, which will tell the interpreter that we actually want to execute a bash command rather than use the notebook. And this can be very useful, for example, if we want to check quickly the checksum or the hash of a specific file. So running exclamation mark md5 sum test.txt, which is very likely to change significantly if we only slightly alter the content of the test.txt. Here, it's changed from d8.49 to But then if we change it back again, the contents has restored. So the only thing that changed was this tool which was added. And I'm mentioning this because it will be useful later on the talk when talking about the patient's data. And finally, quite often, but again, this depends on the kernel that you are using. So we can execute bash in a different way. We can also, yes, the GitHub, but I will commit to it in about 15 minutes from Jupyter Lab to demonstrate how you can use GitHub from within Jupyter Lab, and then it will populate. So just bear with me for 15 more minutes, and it all will be available. So here in this example, JS is a shortcut for abbreviation for JavaScript. If we remove the comment and run this cell again, it will display just one, because that's when developing analysis to be as easy to write as possible, because we want to complete it quickly, and we also want to and please vote for and more challenging on the Discord if you are there, writing your own code or reading the code which is written by please just use reactions to the messages that I shared. It will be a great experiment. So what are you find more challenging? Activity, if you could try to give me a thumbs up on the notebooks channel if you find that writing your own code more challenging, or reading the code written by someone else. Just give a thumbs up, please. That would be very useful. Oh, thank you. I see 3 thumbs up for reading code written by someone else as being more difficult, and 1 heart. Okay, 4 thumbs up. So yeah, it's great that we all agree that reading someone else's code is more difficult. But then the person you are today might be a stranger to the person that will be reading the codes. And whether you are a PhD student as me, or just a postdoc or a master's student, it may be that when you will be reading your own codes after some time, you will not know the person that has written the code. And this is the main thought of what I will be trying the advice that I will try to give you today. So more specifically, in scientific research, we try to develop projects which are easy to maintain, easy to understand, easy to reproduce, and unfortunately also on track to meet the deadline. And notebooks are great help here. However, they are only good for two applications. So this is exploration and storytelling. And you will find lots of posts online telling you why you shouldn't use notebooks for any other purpose. For now, just trust me, those two are the most important and beneficial use cases of computational notebooks for research. And in good reproducible research, you need to tell the story of data exploration. So basically, you could say that there is just one application of notebooks which is telling the story. And this can be the outliers identification, it can be telling the reader what is the distribution of the data you're looking for, or how you matched entities, different genes, different proteins, or how you retrieve data from different databases. However, the process of data exploration is not easy to represent as poetry or prose, and that's because we are often using interactive features like the widgets that I showed you before, or we are moving repeatedly across the code base in multiple iterations, improving what we have done because we find more outliers and we need to iterate again. And we often have many simple plots which are not exactly something that is very interesting to discuss every single time. So then this is a process that needs some additional work, but I think that I may be able to convince you that if done right, it will be beneficial, because in a very different way than if you were to do that using interactive exploration and just noting down the results, you enforce the reproducibility of the key decisions. If you use computational notebooks as your lab notebook. So if you first find out an outlier using an interactive feature and then create a static plot to describe it, you can be sure that it will be there and you won't have to find it again. And if anyone asks you, why did you exclude this particular observation, you can just point them to this plot. If there are any additional data or any changes that needs to be introduced, you can easily rerun the entire exploration process using the code that you've written and compare the results side by side, which is often much more useful than you might think of. And finally, if it's written as something which is meant to be read by others, you might be lucky enough and have other researchers in you who will go and check your work when you ask for that, which is obviously great because there is much less chance of publishing a mistake if someone else will be willing to go forward by that. But some people use notebooks to other aims. One of those is to auto-generate reports. The use case in business data science world is to create repetitive reports of similar structure from a template for different customers, which is very different in biomedical research, where by nature everything is unique or novel, or at least within so where you store the outputs, so where you store the results of execution of every cell, visualizations produced are beneficial to the reproducibility. Storing this output, generated reports, may not be so useful, and you will find people on the internet telling you that you should not be storing your. It's distracting and it makes differences more difficult to spot, but it really depends on the use case that different people, for what they are using the computational notebooks. And I would just argue that research world is different from the business data science world. Then some people try to input the data. You can even use Excel if you like, but probably storing this in a CSV file or TSV file would be a better choice. I will have another slide on that. And some people try to create, to enclose entire code within the notebook, which again is possible, but probably you would be better off if you move all the classes and functions. A rule of thumb that I use, but to a separate dedicated file. Some people even decide to store videos or high-resolution notebooks, allowed to embed. However, if you include a video within the notebook and save it as a single file, it will load in size, and downloading a notebook which is 500 megabytes is not something that many users so if not storing those in a notebook, then where data in to track, so comparing the differences between specific versions will be easier when using tab. Use computational notebooks. They are not the ultimate solution, and there are specialized programs that applications. And this is just a reminder that if you can choose between Excel files and other formats, please do not use Excel, because Excel can be very error prone, because you can include color coding, you can include multiple spreadsheets or multiple different tables within one spreadsheet, and it can cause lots of different issues with date time conversion or gene and metabolite names being converted to dates or times, which is not what you would want, because it most likely, if not noted, will result in the gene or metabolites being not mapped to the appropriate entries in the databases when being read. So for code, it's better for you to store code in dedicated files because you can write unit tests for this code. And I will come to that back later. You can reuse that for other projects, and it's not necessarily easy to reuse notebooks between projects. And this also encourages you to properly document the code because it no longer lives together with the text that you have in your notebook, so you need to prepare a specific documentation which will help you. And also, if you open the code file in any standard editor, for example, Python for Python, it will give you much better support of language-specific features and different code intelligence functions, for example, highlighting errors or refactoring, so changing names, renaming specific variables without breaking the code. So that will be very useful if you want to modify the code later. Finally, what I'm trying to get at is that notebooks are there to tell a story, and if you have too much code, it's likely that no one will understand or will be willing to read that at a critical or deeper level of understanding. It's like with mathematical equations. If there are too many mathematical equations on slides, people are just starting to ignoring them, and you need to cater to your audience and limit the number of complicated code functions which are visible to the reader, depending on who's the reader, obviously. And I think that I don't need to explain why keeping videos and images in a separate media folder is better, but it only concerns high-resolution images. Keeping plots, I think that it is okay. So if you already started using notebooks and have concerns about moving your code out of the notebook to a dedicated file, here are some possible solutions. So if you want to make the reader informed on what exactly is happening, you still can show the documentation of the function. So let's move to an example. Let's assume that we have a function which adds two values. So there is A and B. And this function has a short documentation, as it should. Here we are just adding the integers, but we could be also adding floats. But because I decided to only add integers, it should be also mentioned in the documentation. Perfect to store this function in the notebook because it is very short. Let's just assume that we want to move it to a separate file and create a new file. And call it operations. Let's input it. And here it. So if we use a single question mark, we will get the back displayed together with the signature and location of the file where it is defined. It's not enough. Second question mark. And it should become completely clear what this function is doing if it wasn't already. Then if you want to only have the documentation, in Python you can use inspect. Which will display the text source alone from using a similar function which is called get. Find that you have parameters that you depend on, which are defined in. You most likely still want to move your function to another. Important because if you depend on other variables, it becomes increasingly which variables you refer to and what they are. Not an argument against moving things out of the notebook. It is actually an opportunity for you because you can explain those arguments better. You can define a new name for the argument, which will explain what this parameter does. A docstring or any other form of documentation as well. So then you might think that it's quite repetitive if you are only using this argument. If this argument would be the same for every use of this function. At 5 to every number, so if we had to, if we had different varying and the other number would be constant, it would be always 5, it might seem very repetitive to do that this way. So there is something which is called partial, and in Python you import it from module called function tools. And you have another opportunity to name things. So now you can create a partial which takes function at, sets the b to be always 5, and we can call this at 5. So we'll be always adding 5 to any that we get. And it should work equally well, but it will be slightly more verbose but possibly also slightly easier to understand. And a very similar function is also available in R, although it needs to be installed. So here is the name of the package that you'd need to have, but it wouldn't be too difficult to create this function on your own if you are using R. Now a little bit more advanced topics about reproducibility notebooks in biomedical research. There is some law regulation in the States, which is a federal regulation, and it needs to be considered by everyone who's working on clinical trials, because it defines how the electronic records should be handled and what are the standards for software which is used for any submission to the FDA. And probably this is one of the most commonly considered standards. I haven't heard of any other international standard which would define how you should create code, how you should conduct research if you aim to submit an application for drug approval or anything similar, like biomarkers. And it defines and it requires that the good process of conducting such research or trials should include validation of the code, audit trial, and record retention. So by audit trial, we mean that every step of the analysis should be recorded with very detailed timestamps, and often it should be tamper-resistant or foolproof. So no one should be able to modify these records, and those records should be stored in a secure way for a certain amount of time. So if you haven't heard of that, you're probably not required to obey by these regulations, but because those are one of the most strict regulations, and if you try to strive for the best research possible, you might want to consider and learn about those, because these are the industry standards. And you will find that some of the commercial software does implement the compliance with different requirements of this 21 CFR regulation. So, for example, a tool for metabolomics analysis, which performs PCA and PLS and other advanced analysis, which is called SIMCA, does offer a very good audit trial mode, which makes it impossible to change anything, to delete the history, because everything is from the moment of enabling the audit trial to the very end is recorded in a permanent way on the computer of the analyst, of the researcher. So just before going back to notebooks and how it relates to notebooks, I just highlight that it might not be possible for open source companies to develop high compliance with these regulations, while for-profit companies and which develop closed source software are able to achieve that, and that's why R, for example, or Python performing clinical trials analysis after clinical trials. But then a very good recently done by the R-Core Foundation, and they developed a document that complies with the validation requirements, and they also mention the audit trail as in a bit vague way, because they describe that really say they try to avoid creating specific description of how to do that, and definitely anything that you would do to just generate timestamps according to this document would be sufficient to satisfy the compliance with audit trail requirement, which is different from what the commercial standards and commercial implementation of these requirements are. Tidyverse for such but then it's not bear with me for a moment. Okay, so here is a missing slide. So as it turns these requirements by default, because I don't know if you noticed, but I can execute cells in any order. One, and it has execution order 32, and cell with 2 has execution number 33, but now again and again, and the information of which cells were executed in which order in the meantime. So while some books interface and force you to execute the code once you want to check, this is not strictly true for others, and the history is usually not saved, so that the entire process of developing the notebook in your mind, and that's why you often need to use dedicated version control solutions. But you can still use extensions which add timestamps to notebooks. Force yourself to execute your notebook to check that the code that you've written in various order, executing various cells, will also work come to be. And just to give a quick demo of that, we have written an add function previously here, and if I were to call this function at this place, it would work right now because it was already imported. But if I were to restart the kernel and run all the cells up to this moment, which is happening right now, as you can see by this black circle. So kernel is busy. I need to execute this cell now. It will say that the name add is not defined because it is only imported here. So I will remove that and just highlight that in the new version of Jupyter Lab, there is this button, restart the kernel and rerun the whole notebook, which is very useful to ensure that you don't have any errors after executing, after modifying your notebook. And I will just press it right now. So then if you are using R Markdown, then you can use session info. You can add this call to this function at the very end of your notebook or R Markdown document, and it will display the current session, the packages that you have attached, and the versions. So anyone who would want to verify why there may be differences between the results that they obtained and that you obtained, what might be the reason for differences, because it might be coming from differences in the package versions. And maybe another Jupyter specific thing is that when using Markdown, you can use backticks to create code or to create text which looks like code. Okay. And onto some more concrete, more specific advice on working with notebooks. I would highly recommend you to start every notebook by defining its scope. So providing a description of what you will be doing and aims which can be verified in a smart way. You could say, so whether there are any specific outcomes from these notebooks, any data frames to be produced, any test results to be obtained. And then if you have multiple notebooks which have slightly overlapping scope, for example, you have different molecular analysis and you perform this analysis for different sets of patients, you might want to state that this notebook is only for patients from center X, another notebook is for the patients from center Y, and there is a third notebook which will look at the combined analysis of the molecular data from both centers, and none of these previous notebooks should attempt to analyze the aggregate data from all the patients. And to give you an example of that, there is an example project with a very brief introduction and scope specification. So I usually do something like that. So describe what is this notebook about, what are the aims, and in this case it would be providing a clean long format data frames which would include patient demographics, medication use, and subject sample mapping. Then, if you have multiple notebooks or larger analysis that requires you to move one step at a time. Beneficial to separate data preparation step, data exploration, analysis into separate direct, as demonstrated. Here we would have prepare data, explore covariates, and survival analysis. So the structure of an example project to demonstrate how you can manage your notebook in an orderly fashion. If you prepare data, I would encourage you to one notebook, because this is the easiest to later go back and the files that you are working on or get updated by other collaborators, and you would have to find in which notebook you analyzed. Or so having that in the data preparation step, one-to-one relation, can be very helpful. In the data preparation steps, along with other code-heavy tasks. So anything which requires lots of code just to get the data into the right format should be contained within the first data preparation step. Then onto the data exploration, you might group the related data groups together. And by data exploration in this example project, we would have explore covariates here, for example. If we were looking at a study of patients who had different metabolites measured, data available, and we would several notebooks just to explore the different data covariates, demographic characteristics, and perform quick QC checks, and then have additional notebook specific omics in this particular example. And this slide is cut off, so let's go back here. For the final analysis, you should generally try to make your data with a little bit less text, and you should have as much text and explanations as possible in the final steps, and as little code in there, because that is what makes it easier to read by code literate, as you might be after attending all of these seminars. And you also can move some functions, some code that you might find yourself repeating at the beginning of each notebook, and run it with this command. So created a file notebook setup and have some very useful functions in there. So I created such a file here, and because this is Python notebook, I very often use matplotlib and ggplot. So I would load it here, that I frequently use, and in any notebook, I would just run it. Kernel that I use. So this is the default Python kernel. It wouldn't work for R, but it's something that you can accomplish with other means just by contains the other ways of initializing the notebook. But I would just advise you not to put too much into this notebook setup, or whatever you call this, because then it would become difficult for the reader to understand why, from where the specific variables are coming from. Another potentially interesting thing is the ability to set seeds in such an initialization file. It's obviously not always a good thing to define seeds at the very beginning, but it does help to produce reproducible, replicable graphics with all the elements placed in the same at the same positions if there is any random element to the algorithm that defines the layout. And the most important part of this seminar will be the 3 tools that I wanted to introduce you to, which should help you avoid any mistakes. So the first tool, which I hope will be explored later on, tests. And tests are different from validation, so they only check if the code that you've written performs as you would have expected. And they are not validated by the entire team of experts who would check that it is indeed that what the code should do, so whether your expectations were also correct, if all the corner cases are covered. And at the very minimum, it prevents you from breaking the properly functioning code when trying to improve it. So just very quickly to demonstrate how you would write tests, let's assume that we have a function which divides two numbers, and it should be fairly easy. To define, and we could create some test functions which would verify that this is actually what we expect the function to do. So this is not the best example because I have had a peek. I have checked the output from this function first. So let's just run this and see. So the assert construct in Python, and there is also a similar construct in R, similar keyword, will raise an error if the value on the right-hand side is not true. So if we have false here, it will raise an assertion error. But if we had true, it will just pass OK. So we can now create a function which will group together several different tests maybe let's just call it divide. And in the other test case, we could check if 10 divided by 2 is 5. And this should be fairly straightforward. We'll just call this function, and if it passes, everything is great. So we wouldn't usually write this function in a notebook. This is just to demonstrate how to approach that. Those functions should reside in a separate directory. So in the example project, it's a directory called tests. And here we would have a file called test operations or test at. And okay, so there is a question on test. So we would import the divide function. So that's I'm seeing an error, and I will explain why. But it tells me that the divide function is not defined here. So I would have to import it, and if I tried to import it from the notebook, it would require quite a few hacks and workarounds which are not recommended. So move it to the operations.py file that I have created here. Then import it right there. The test functions specific programs which would go over these files and run these test functions for us and provide very nice reports. I will not cover that in detail because this is not the main focus of the talk, but I just wanted to highlight that tests should not usually be contained in the notebooks. They should live elsewhere, but are equally important to be developed together with the code that you write, even if it's in the notebooks. And you can write it as I did here in the notebook and then just to the test files. As to the question that showed up on the notebooks channel, when to write the tests at development stage or after function is written. So there are various schools, various approaches. There is something which is called test-driven development, which writing the tests and only then writing the actual function. You can use a mixed approach. You can try to fast write it, but usually it would lose from that, because writing tests is often seen by our brain as something boring, which it isn't. Just adding more onto this topic of you might want to have a very specific expectation as what should happen in this case, and give it some thought. What do you think that should happen with your program or your code if you think arguments 1 and 0 as shown right now? So if you just leave this function as it will raise 0 division error, which is how it was decided in the Python language that it would work, but it might not be to do with a warning for the user. So you might want to write a test like that. So when there is division by 0, we want the function to return a string rather than raise an exception. And it should tell us that it's a warning. It's not a warning sense in a strict sense, because there is a specific mechanism to warnings in Python, but for the sake of simplicity, we could catch this error here. No, not this language. And if we were to run this now, it would still raise an assertion error because I have not executed this cell. So we can see that it's actually 0 division because it's propagated. Now I will execute the divide function. I will also execute the earlier tests to check if they still work. And now this test also passes. So another tool that I wanted to highlight is linters. So these wavy or dotted lines that you see here and the warnings are basically what linters do. They tell us what is wrong with the code. It is not necessarily an essential thing if you want to code, but from my experience and do, and you should configure your editor to support such warnings, because if I try to use a function, so here it no longer highlights me to enter 2 blank lines, so I did that, and now it wants me to remove this white line. But if I made a typo and it didn't work this time. I will come back to that with another examples soon. Let's go back to the slides. Various linters will either highlight your mistakes, typos, or just teach you good style for a specific language. And finally, diffs, which is a very important component of using Git for versioning or any other versioning system, because it allows you to catch any unexpected or unintended differences between versions of your files when you create a new version. So comparing notebooks is not something which is very easy if you try to compare the Jupyter notebooks, because those are encoded as JSON files. And let me just show you what does it mean, because I mentioned this term and have not explained it properly. So if I open the notebook with a simple text editor rather than with the notebook editor, I will see a structure like this, which is not necessarily easy to work with, but it is very good for the programs, for other software to process that, which is different done with R Markdown, which is easy to work with as a plain text, as Markdown document, and not that easy to work with if you want to process it with different tools to create more advanced notebook interfaces or tooling to work with notebooks. But there are dedicated solutions to compare notebooks, and about that in a moment. So the free tools are not sufficient to make your code perfect, but it's definitely a good step forward. And if you are convinced that using notebooks is something that you would like to do, then I would strongly encourage you to embrace all of this free as a good step forward. Let me check. Okay. So in addition to developing tests in showcased, you could also execute entire notebooks, dedicate entire notebooks to tests, or choose any different ways of doing so, because there is not so much standardization. I encourage you to develop the tests in or to store the tests in separate file rather than in notebooks. And this is partially because you would start mixing tests and storage. It might be that having the notebooks as a tool to tell the story about the data would be more beneficial. Different use cases and confusing the readers, especially if they are just biomedical researchers and not programmers, or not biomedical researchers who code. And if there was a research engineer called research software engineer, and you may be able to get help from them at your university, this is something recent, but very important, and they may be able to assist you with transition to notebooks if you ask them, or with rather than have to open each and every notebook to find the tests that you were writing somewhere in between your code and prose. Add on to that, in addition to, and that would include, for example, checking that everything is as expected, for example, the patient's age, to highlight statistical models of just when coding, which again, the fact that your data meets the assumption, passes the checks which are required for a statistical model, it might not be the case if you 6 months' time or 1 year's time after the data has changed. So it's very useful to have a short code like assert something, assert, let's say, age is and here H is a series of and it passes without any expected. Possibly also either that those things are important or that is what is expected. So about linters. In Python, there is something which, if you have a Python interpreter available, could you please type import this and see what happens? If you haven't already seen that, I hope that some of you can smile on the upon seeing the output. So this prints out the XenPython, which describes what code is considered good in Python. There is a very strong culture of writing good code when writing in Python. As you might say that it's not always the case with languages without naming, which will be reflected in the warnings that you will find when using linters, some of which will be very aggressive and require you to modify things that you would think that are okay. So for example, let's just divide two numbers, and you will see that the linter, which is called bytecode style, produced an error because it would want me to have a space between the operators. And small style issues like that and larger issues related to how you write code might be highlighted by these programs. So those are basically programs that help you write better code. And a fairly recent addition is called MyPy. So it does check whether the values that you pass to your functions are of the type that you specified to them to be. So in the add example here, I specified that I only expect integers. And if I had MyPy installed and specified, I think that I do not have it installed currently, but if I specified something like this, which is floats, it should highlight that this is not what was expected. I will post also instructions on how to install the static type checking extension to linters, to various linters, online in this repository after the session ends, because I just didn't have enough time to complete that before. So if you want to use all of these features in notebooks, it might come to your somber realization that it's not yet very well developed for notebooks, which is partially because notebooks are much more complex with different cell types than simple text files. with code. So for example, this is a code, simple file with code, and it is much easier to check syntax and code within this file than it would be in the notebook. So the environment of coding assistance tools for Jupyter notebooks is still in early days, and there is some work for Visual Studio Code from Microsoft. There is some work that I am engaged with in which is called Jupyter Lab LSP, and this is basically what provides the checking in the notebook that you are seeing. So all these underlines which would show up, for example, here, would not be present if in the default installation of Jupyter Notebook, so Jupyter Lab. So you have to install an additional extension to get this benefit. For R, there are also many different tools which have slightly different scopes. So linter is error checking, and styler is more about checking your style, as you might have guessed, and they are by default available within the RStudio. However, you can also have them Jupyter notebooks after installing. And on to the final bit about diffs. So yet another extension generate diffs between notebooks as well as files from within the Jupyter Lab environment. Show that on an example. So there is an extension manager which usually, and you have to enable it, because if you install extension on websites, then it will take care. But after installing something which is called Jupyter Lab/Git, the lab team, you will get this extra icon here, which allows you to operate Git from the and having to go to the terminal and add the files manually. So I will just using this plus. So all of that should be shared with you. Okay. And I will just commit the version line, so you should be able to see all of that in a minute online in the GitHub repository link provided. Okay, and let's make some changes. So for example, let's remove this. And this, and this. and save the file. So after I save the file, it has shown up in the changed, and I can press this button to div the file and create a report of the changes that were introduced. So if I scroll down to the place where I deleted cells, I will see that they were indeed deleted, and this one was deleted as well. So because they are in red, they are deleted. But then there is another button, maybe confusingly, which does basically the same, but the display is a little bit different. Let me just add maybe one cell with some text to also demonstrate how an added cell works and rerun this cell. So there is a difference, and I will save that and diff. So with this tool, it is calculating the differences, but the cells, we didn't see the cells showing up because all the cells which had no differences were hidden by default, and we only see this cell because I ran it again and the execution number has increased. It is possible to disable this function so that the changes in the number, in the execution order number, are ignored. And then we see that these 3 cells were removed and this cell was added. So this is a very useful thing, because then you are able to control what changes are being introduced, and this is much better than comparing the JSON representation as would be default on GitHub at the moment. Okay, let's catch up on the slides. So because we have still a few minutes, there is a wordy slide on dealing with patients' data. So I just assume that everyone who is dealing with any patient's data is using anonymized data and is kept locally in a password-protected file outside of the repository. But I just highlight that it might happen that you will print out the entire data set in your notebook just to explore it, just to have a look. And it might be that even if the data is anonymized and it wouldn't be a huge deal if it was committed to the repository, which is private, then you might still want to avoid that, and using diffs is a great failsafe to check that you are not including any data which would be better kept private. Then notebooks are particularly prone to potentially revealing any data which could be already anonymized, in the way that there is no easy way to connect the data back to the patient. But if we stop looking at the entire data set and at the summaries and look at the outliers only, if you think about patients who are likely to be outliers for specific analysis, then you might find that patients from minority backgrounds are recruited at smaller experimental sites, smaller hospitals. And in this example that I provided here, after, for example, many pregnancies, they are by definition more likely to be outlier because those characteristics are not as frequent in that population in the study population, and then they are also much more likely to be more easily identified. So I just encourage an extra caution when dealing with outliers, when working with patients' data. And even if we say, and if we know that the data is anonymized, if there are other details, at the diffs between the notebooks carefully again. Very strict and conservative approach on what is to be included and what should be better kept from that is not any extra details. To in a very specific case, if you have to display all the data about patients, but you do not want to include that in your notebook because you want to make sure that you are working with the same copy of data, want to see that it changed between the versions. You can use some mechanism of MD5, for example, that I mentioned previously, and demonstrated how to run that in the Python. So while this is not guaranteed to be unique and not guaranteed to not have a collision, it is highly unlikely that you would get any collision when changing the data. So this very short text that you retrieve from MD5 sum function on Linux or MD5 command on Mac to version of the data that you are looking at has changed. And then if you will work with the data as long as substantial probability or likelihood of getting a collision, then in thousands or millions of years, you will have probably more pressing issues to worry about of the checksum, as noted here. Okay. Just additional recommendation on passing the data around. If you are dealing with RDS in R, to do that because there are many small Python, some of which are specific to R. It's compatible if you store the data with a newest version of R. You can use dedicated and version the data. And this is mentioned on the slide specifically because when working with larger projects, it might be beneficial to have the data at this point right now as connecting different notebooks together, because what I showed case before, so the structure of the project, exploring, showcasing this notebook, for example, committed to the repository right now, is okay for a medium-sized project. But then if you have a very large project with lots of notebooks, an ordered list of all notebooks, the exact steps in which you have to run specific notebooks to achieve what you want. And you might want to instead have a data management system where the input and output files are nodes and notebooks connect them, which allows you to create automatic execution solution for large projects. So, for example, using Papermill and walking over a directed acyclic graph in deduced order, you can achieve a system like this, which for my use case is called NB pipeline and allows you to reproduce, let me showcase this here, to both run several notebooks and also compare them and check whether the exact calculations and results are easily reproduced in those notebooks, which are rerun again in a separate environment. And that would be all from me. Thank you very much for staying with me, and I hope that it was beneficial for you and for your future projects, which might grow in size, I hope.